MD Anderson Cancer Center has a reputation as the top cancer hospital in America with a mission of making cancer history. It's where Dr. Stephen Curley is carrying on the vision of John Kanzius to attack cancer cells and tumors with radio frequency waves and leave the good tissues unharmed. Trials on animals are producing remarkable results.
John always envisioned that there should be a better, less toxic way. And the results have actually been so good at times they're a little scary. I worry that people will believe our results because we really don't see any damage to normal organs or tissues in these animals.
Dr. Curley invited us into the 3 laboratories in Houston where his team of 18 researchers, all with diverse expertise, is targeting cancer in their ongoing research.
We have chemists, we have physicists, we have engineers, we have physicians. Actually, we have fellows who are working in Dr. Curley's lab. So we are bringing up physics, chemistry, math, and biology, everything into one table, and we all discuss how do we solve this.
I'm here in the 4th floor lab of the MD Anderson Cancer Center. The focus here is on the cancer cells themselves and the nanoparticles. And there's also some pretty expensive equipment here, like this flow cytometer, going for about $400,000.
And yes, it was purchased by the Kanzius Foundation. Researcher and lab manager Warna Kalua-Rachi uses its laser and color technology to track how many cancer cells are killed after treatment in the Kanzius radio frequency field.
69.9% of cells are viable after the first round of RF treatment.
So we have here an example of 10 nanometer gold nanoparticles. The reason it looks red is actually because of a property called plasmonic resonance.
Stuart Core, nanoengineer, is the expert on the very tiny metal nanoparticles used to attach to cancer cells and heat up under the radio waves.
So in the case of our cancer therapy, we want a nanoparticle that absorbs a lot of energy and converts a lot of it to heat.
And the key is getting those particles to attach only to the cancer cells.
So we have to identify on the cancer cells something that is abnormal, a protein or a sugar, something that's on the surface of the cancer cell that is not on the surface of normal cells.
Research fellow Sophia Punsavath is at work in this protected part of the lab where cancer cells are grown, manipulated, and incubated for the ongoing tests. Dr. Curley says in 2 of the toughest kinds of cancers, by adding antibodies or proteins, they are answering the key question.
Can we get the nanoparticles to go to the cancer cells but not to normal cells? Again, we don't want the nanoparticles taken into normal cells. They would also be damaged by the treatment then. And so for pancreas and liver cancer, we believe we have the ability to do that.
As the cell research advances, so does the design of the Kanzius RF machine, all in the push toward human trials. Lisa Adams reporting.